we are talking about some details of chromosomes and cell division. We understand that in a cell there is a nucleus. Within the nucleus you will have some 46 chromosomes, 23 from mother and 23 from father. Total 46 chromosomes. Now when you arrange them, you will find that there is 22 pair of chromosomes which are called autosomes, and there is one pair of sex chromosome. If it is a female, then she'll have XX chromosome, and if it is a male, it will be XY chromosome. Also understand that when we are talking about chromosomes, the 23 pair of chromosomes, one is from mother and one is from father, and they look alike. They are called as homologous chromosomes. So you may see that there is a taller set of chromosome, there is a short length set of chromosome, and like that will have 23 pair of chromosome looking alike, called homologous chromosomes. In a chromosome, you will notice that there is a centromere which is holding the short arm which is P and the long arm which is Q together. Chromosome is nothing but a double helical of DNA, and before division, before any cell division, it must undergo DNA replication, doubling of DNA, which happens in the synthetic phase of interphase. When a chromosome is going to enter cell division, before that it will double the DNA, you will see now we have sister chromatids. These are called as sister chromatids or duplicated chromosome. If this is n amount of DNA, then it will be 2n amount of DNA. So this you just keep in mind, and we can proceed further by drawing a diagram ourselves as well. You see, when we are talking about the cell, in a cell there will be the nucleus, and in the nucleus there will be chromosomes. And if you are talking about the chromosomes, then you will see that one is maternal and one is paternal. In those chromosomes, this pair, homologous chromosomes, you can have 23 pair of chromosomes and they look alike. If you say they are looking alike, or they are called as the homologous chromosomes, what they do is, before they are going to undergo cell division, they have to duplicate their DNA. So it will be like this after some time, that you can see what is called as the sister chromatids. Normally, you have to understand, we are diploid organisms, 2n, where n is the number of chromosomes. Now when you say that we are 2n or diploid status, which is n is number of chromosomes, you can also mention it is 2n and 2n, which is capital N. Now what is that capital N? That is the amount of DNA, and you have to understand that I will always get 1 unit of DNA from my mother and 1 unit of DNA from my father. So any cell in my body, skin cell, blood cell, bone cell, hair cell, or whichever cell, most of them are diploid, 2n, 2n. But before the cell division begins, that duplication happens. So 2n would have become 4n. So that 2n becomes 4n before the cell divides. And why does that happen? Let us see the details. You will notice that when you are talking about gametogenesis, which is formation of the gamete, there is a primary gametocyte, which could be a sperm or oocyte, which is diploid. And then it is dividing. Say this is meiosis. Meiosis, two-stage process, meiosis 1, and then there is meiosis 2 also. In meiosis 1, the primary oocyte will enter meiosis 1 and then give you what is called as the secondary oocyte. And the secondary oocyte is then going to divide into the final oocyte, or what is called as mature oocyte. In case of a male, you will see there is a primary spermatocyte which is becoming secondary spermatocyte which is undergoing meiosis 2, and then you will have the spermatids. Then you will also notice that here, as you find, a male will have sperm and a female will have oocyte, will fertilize, and then a zygote is formed. The point is, meiosis is a cell division where the homologous chromosomes are separating. Maternal and paternal chromosomes are separating. So this cell which you find here becomes haploid. And this haploid cell is having duplicated chromosomes, the sister chromatids, and the sister chromatids are going to separate during meiosis 2. So during meiosis 1, the homologous chromosomes are separating. The next cell, which is the secondary gametocyte, become haploid. And after that, the sister chromatids are separating, and then these are all haploid gametes. The gametes are actually haploid. We'll have to look at more details for this now. Understand, when we have the zygote, it will have two nuclei, maternal and paternal nuclei, and it is dividing to give us structure called blastocyst, having some inner cell mass and some epiblast. Those epiblast cells are dividing to give you primordial germ cell, which is the first sex cell of our body. And these primordial germ cells then keep dividing gonocyte into the gonia, then into primary spermatocyte, primary oocyte, then secondary, and then you will have the mature oocyte. In male it will be the spermatocyte. Let us make this diagram ourselves now. You have to understand that we are talking about the zygote here. And when you say it is the zygote, it will have two nuclei. One is maternal and one is paternal. Now, if you say there is a maternal and paternal nucleus in the zygote, fine. What is zygote doing? It keeps dividing. It is a single cell. It will keep dividing and give you certain further stages, and one of that stage is called as the blastocyst. Now, in the blastocyst, you will find that there are some epiblast cells, and these epiblast cells are the one which are going to give us the first sex cell of our body, and that first sex cell of our body is called as primordial germ cell. Now, this primordial germ cell has the capacity to become male or female type of gametocyte. In the beginning, it will give us what is called as the gonocytes, and those gonocytes are going to become what is called as the gonia. You can say gametogonia, and that gametogonia can further give us the gametocyte. Now, when you say it is going to give us the gametocyte, it could be a primary gametocyte, or next is the secondary gametocyte. And also remember, this could be a female, then you will say oocyte, and if it is a male, then you will say spermatocyte. So, as you can understand, in the beginning there is a single primordial germ cell, but it can form a male or female depending upon the fact that whether you are XX or XY chromosome. So if I am a male, then spermatogenesis. If it is a female, then it is oogenesis. These gametocytes, which are primary and secondary, finally they will give us either the spermatocyte or they will be giving the oocytes there. Now, as we can make out, oocyte from the female mother and spermatocyte from the father, they unite to form the zygote, and the diploid status will be established. Let us move on to some further details. You will notice that cell division, when you talk about, there are two types, one is mitosis and meiosis. Let us make a diagram now for the cell divisions. See, we have got a primordial germ cell, and where it came from? It is coming from epiblast cells. So it is the epiblast cells which will give us primordial germ cell. Actually, Epiblast cells are going to form what is called as primitive streak, and in the primitive streak there will be the primordial germ cells developing. And these primordial germ cells which develop in the primitive streak now migrate towards the genital ridge, because at the genital ridge they are going to give us what is called as the gamete. The process is called gametogenesis. So when you're telling gametogenesis, it is the formation of the gamete from a cell which is called primordial germ cell. Sometimes there may be a problem in the migration of the primordial germ cell towards the genital ridge, aberrant migration. And if we have got a case of aberrant migration, there can be tumors. And what do you call those tumors as? They are called as germ cell tumors. So we are mentioning, if the primordial germ cell is having a problem, some aberrant migration towards the genital ridge and going to some other place, then there could be germ cell tumors. And these germ cell tumors, like sacrococcygeal teratoma, if you're talking about this sacrococcygeal teratoma, a question comes, why are you having it? Because there is something abnormal with the primitive streak. The primitive streak generally will disappear. And what if it is persisting? If it is persisting, then it can give you tumors. And which tumor? That is germ cell tumors. Okay. Also notice that sacrococcygeal teratoma, which is maybe coming from primitive streak or primordial germ cell, is going to have all the three germ layers. But why it will have all the three germ layers? It is having ectodermal derivative, it is having mesodermal derivative, and also endodermal derivatives. But why? Because this primordial germ cell is a pluripotent cell. It can form any of the three germ layers. Now let us move further with the gametogenesis. There are two cell divisions involved, which are mitosis, meiosis. Now if you say gametogenesis will require some cell division, which are mitosis and meiosis, what is the difference between mitosis and meiosis? Actually, mitosis is equational division. It will not change the chromosome number. If mitosis is equational division, do not change the chromosome number, then what about the meiosis? Meiosis is actually a reduction division. In fact, meiosis 1 and meiosis 2. And it is meiosis 1 which is a reduction division. What do you mean by reduction division? Means it will reduce the chromosome number to half. So it is meiosis 1 which is a reduction division, reducing chromosome number to half. And mitosis is equational division, does not change the chromosome number. What you have to understand is, in the beginning, we were having one chromosome from mother and one from father. So it was actually 2n, 2n. When you say it is 2n and 2n, then what is happening in the gamete? It is half the number. If you say it is half the number, means you can have either maternal or paternal chromosome. Yes. A gamete can have either maternal or paternal chromosome. Actually, it will be a mixture of maternal and paternal chromosomes. 23 from mother and 23 from father. But it is a mixture. The hair color may come from mother side and the nose size may come from the father side. So this is gamete, which is N and N. So if it has become haploid from a diploid status, when does that happen? That happens during meiosis 1, which is a reduction division. Now if you say like that, fine. What are the further details then? Let's move on to the further details. When you were talking about the germ cell tumor, you said one name, that is a teratoma. See, this is a baby who is born with the oropharyngeal teratoma. Normally, primordial germ cells should go to the abdomen region, because in the abdomen region you have the genital ridge to form units. Now if you say the primordial germ cells should go to the abdomen region and form the gametocyte in the genital ridge. What if they have migrated towards the oropharynx? Then oropharyngeal teratoma. Or sometime, the primitive streak is supposed to disappear. But if primitive streak is persisting, then primordial germ cells are persisting and they can form tumor. Like this is a tumor at the tail of the baby, and sacrococcygeal teratoma. As you are moving further, let us draw a flow diagram. The primordial germ cell is going to give us the gametogonium we have seen, and it is the gametogonium which is going to give us the primary gametocyte. And we said that primary gametocyte could be male or female. If it is male, then you will say primary spermatocyte. And if it is a female, then you can mention it as primary oocyte. Now understand, these conversions, when you are talking about the primordial germ cell becoming gametogonium, becoming the primary gametocyte, is all happening by mitosis. This all is mitosis. But beyond that is meiosis. If you say beyond that is meiosis, then which cell is entering meiosis? The cell which enters meiosis 1. Of course, meiosis 1 will come first. The cell which enters meiosis 1 is obviously the primary spermatocyte, or could be the primary oocyte. Also remember one thing, females begin oogenesis quite early as compared with the males. Spermatogenesis begins after puberty, but oogenesis begins before birth. When the female baby was in the uterus, in utero, oogenesis has already begun. So oogenesis begins quite early, and the baby was still in utero. You mean to say the primary oocyte would have already entered meiosis 1 before birth. Yes, before birth only they are in meiosis 1. That is what you have to remember. Now as you move further, you will first compare between mitosis and meiosis. I told you mitosis is which kind of division? It is called equational division because it will not change the chromosome number. But meiosis, specifically meiosis 1, is a reduction division because it is going to reduce the chromosome number to half. And as you proceed further, this is the two divisions getting compared. See, it is a diploid cell in the beginning, the somatic cells, and then they are undergoing DNA duplication. Before any cell division, you must have DNA duplication. So if you say this was actually 2n, then now it has become 2n and 4n. Because you have got duplicated chromosome and sister chromatids are there. As you move further, you will see mitosis is going to divide the cell into 2, and again finally there will be 2n and 2n. This cell and that cell also separately. So this is mitosis, and it will not change anything. It happens in somatic cells like muscle cell, bone cell, hair cell, skin cell. But then meiosis is different. How meiosis is different? You see, in meiosis also, of course, when the cell is undergoing division, it will duplicate the DNA. As you see, in meiosis, the duplication of chromosome, which is now you have sister chromatids, there is some exchange of genetic material also. There is a chiasma formation, and in that chiasma formation there is some exchange of genetic material between homologous chromosome, one from maternal side, one from paternal side. They are exchanging the DNA segment. Some genes are exchanged, genetic recombination. And then you will notice that we are looking at meiosis 1. In meiosis 1, the homologous chromosomes are separating. Either this cell will have maternal chromosome or it can have paternal chromosome. And after that, there will be a meiosis 2. In meiosis 2, you will notice that the sister chromatids are separating, finally giving you haploid cells, and which are 4 in number. If you compare, here you have mitosis. The net result was 2 cells which are diploid. But in meiosis, the net result is 4 cells which are haploid. So we want to talk about some more details now. As you will see, we are talking about gametogenesis, male-female. Male means spermatogenesis. Let us first focus upon spermatogenesis. And there comes a question which is asking you about the polar bodies. See, polar bodies are seen in females. In females, the ratio of the primary cell and the final cells is 1:1. You mean to say, if I start with one primary oocyte, finally I'll have one mature oocyte only, because there are 3 polar bodies which are discarded and degenerated. This does not happen in male. Maybe in female the ratio is 1:1 and 3 are wasted. In male it is 1:4. Start with one primary spermatocyte, will have 4 spermatids. So this is one difference which you have to remember. 1:1 in female, 1:4 in the male. Now let us briefly look at the male process, spermatogenesis. You notice a question. What is the false statement about spermatogenesis? Spermatozoa are formed from the spermatogonium. Yeah, of course. Taking 74 days. Yeah, that is correct. Which is the largest cell? You will find that yes, the largest cell is primary spermatocyte, but if you say meiosis occurs in secondary spermatocyte stage, that is not correct. Why? Because you see here, we have a spermatogonium which is going to undergo the meiosis 1. Remember, the largest cell is primary spermatocyte. Now it is dividing first meiosis to give you secondary spermatocyte. And the secondary spermatocyte will then divide to give you the spermatids. And these spermatids will form the sperm. The spermatogonium forming the sperm takes 74 days. That is the point. Largest cell is the primary spermatocyte. That was the point. Here, the homologous chromosomes are separating, and here the sister chromatids are separating. So these are some of the questions the examiner is interested in asking. Tell me again, what is the false statement? We also already have actually known that the answer should be choice number D. But why? Because meiosis starts happening at the stage of primary spermatocyte and then the secondary spermatocyte. So here it should be primary spermatocyte. That's why this is wrong, this is false, and that is the answer. Talking about oogenesis, you'll find that we have got a question asking which cell is undergoing fertilization. The same cell which is undergoing ovulation. And who is that? It is the secondary oocyte. And which stage? It is the secondary oocyte which is arrested in metaphase. Now if you say the answer is choice number D, explain why you have taken that answer. You have to understand that there is a primary oocyte which is undergoing meiosis 1 to give you the secondary oocyte, which is then giving us the mature oocyte, and that is 2 cell divisions. Actually meiosis 1 and meiosis 2. In meiosis 1, the maternal paternal chromosomes separate and there is a release of first polar body, and in meiosis 2, the sister chromatids separating and release of second polar body. When you are talking about maternal paternal chromosomes separating, that is also called independent assortment. Assortment means separation. Why would you say it is independent separation? Because 50% chance that this could be maternal chromosome, or 50% chance this could be paternal chromosome. So independent, it depends upon nothing but 50% probability. Now independent assortment which occurs during meiosis 1 separating maternal paternal chromosome in equal proportions, primary oocyte was actually arrested in prophase 1 diplotin stage until puberty, because only after puberty there will be LH surge, and after LH surge you will have what is called as secondary oocyte, and the secondary oocyte will be then arrested in metaphase 2 of meiosis 2, and then it will be undergoing ovulation. But there is a probability that this secondary oocyte may undergo fertilization, but most of the time it is undergoing menstruation, because fertilization is a rare event. If fertilization happens, then you'll have what is called as the second polar body, and also the mature oocyte. Actually, mature oocyte will be a fertilized oocyte. How? Let us draw a diagram. Let's draw the same diagram now. What I'm trying to say is, we have got a cell here which is called as the primary oocyte, and this primary oocyte is actually arrested in prophase 1. So this primary oocyte was arrested in prophase 1. Which stage? That is called as the diplotin stage. This primary oocyte is arrested in prophase 1 in diplotin stage in utero only, before the baby was born. It was already in meiosis 1, arrested in prophase 1. And how it will become the secondary oocyte? By undergoing meiosis 1. But for that there is a requirement. You must have what is called as LH surge. So once the girl will achieve puberty, she'll start having LH surge. Then only the primary oocyte will become secondary oocyte. So if you say now you got the secondary oocyte here, what else do you get along with secondary oocyte? That is the polar body number 1. Now remember, the first polar body is released after LH surge, but before ovulation. So first polar body is released after LH surge, but before ovulation. And what about the secondary oocyte? The secondary oocyte is now arrested in metaphase 2. So secondary oocyte is now arrested in metaphase 2. Yes, metaphase 2 of meiosis 2. It has already entered the meiosis 2, but in meiosis 2 it was arrested in the metaphase. What it requires is ovulation now. So the secondary oocyte arrested in metaphase 2 of meiosis 2 is undergoing ovulation, and then it is waiting for the sperm for fertilization. And what if the sperm is not available within 24 hours? You have to understand that fertilization should occur within 24 hours of ovulation, otherwise the secondary oocyte will be degenerated and is menstruated. The secondary oocyte, if it doesn't meet with sperm within 24 hours of ovulation, then the secondary oocyte is degenerated and menstruated by the end of the cycle. Fertilization is a rare event to occur, and if it happens, then you'll have the next cell, which is called as the mature oocyte. Now some books will write it as ovum. Ovum term is not mentioned by Gray's Anatomy anymore. It is discarded. It is obsolete term. But still some books will use the terminology ovum. So mature oocyte, which is the other name for ovum, is actually released along with the second polar body. You can also mention here is the polar body number 2. But remember, the second polar body is released only after fertilization. So second polar body is released after fertilization, and this is along with the mature oocyte, but it is a fertilized oocyte. How do you know it is fertilized oocyte? Because it has one nucleus, which is the maternal nucleus, and it has one paternal nucleus also. You mean to say sperm nucleus? Yes, there is seen the sperm nucleus in the cytoplasm. Now, if you say this is the sperm nucleus seen in the cytoplasm, and that is why you are telling that it is a mature oocyte or fertilized oocyte, what will happen when the sperm nucleus reaches the center? Then you can call it as zygote having two nuclei, maternal and paternal nuclei, and then it becomes diploid. Half the chromosome will be given by father and half the chromosome are already with the mother, and they will become diploid. So this is what we have to talk about in more detail. You will notice that when you look at the ovary at birth, ovary will have primary oocyte already there, arrested in phase 1. We are looking at the ovary at birth here, and if you are searching for the cells, you will notice that this is the primary oocyte, which is present in the ovary at birth, and it is already entered prophase 1. Actually, it was arrested in prophase 1. Which stage of prophase 1? Because prophase 1 has certain stages like leptotene, zygotene, pachytene, diplotene. The question will come, you're looking at the ovary at birth, it has primary oocyte, and that is arrested in prophase 1, but which stage? And your answer is diplotene stage. But why it is arrested in diplotene stage? Because of OMI. And what is OMI? It is the oocyte maturation inhibitor. Now if you say it is the oocyte maturation inhibitor, what is this exactly doing? What is it doing? See, this oocyte maturation inhibitor is affecting the cyclic AMP. What it does is it will actually increase the level of cyclic AMP and causing the prophase arrest. But how will I reverse it? Actually, when this raise like cyclic AMP is there, this cell cannot move into the next stages, which is the metaphase or anaphase or telophase. Now, if it cannot move into metaphase, anaphase, telophase, then how do I break the prophase arrest? For that, you must achieve puberty. Now, if you say the girl has achieved puberty now, she will start having LH surge, and when she is having the LH surge, then you will find that there is a change of cyclic AMP. Once the girl has achieved puberty, she is having the LH surge. Now it is going to reverse the level of cyclic AMP. You will notice here there will be a reverse, or you can say reduce the level of cyclic AMP, and then only the cell can move into metaphase, anaphase and telophase. Remember one mnemonic. Your mnemonic is P, P, P. And what is that extension of the mnemonic? When you talk about the extension, the first P is the primary oocyte, and second P, that is the prophase 1, and the third P, that is the puberty. Means you have to remember, primary oocyte is arrested in prophase 1 until puberty is achieved. So primary oocyte is arrested in prophase 1 until puberty is achieved, and after puberty is achieved, the girl will start having a LH surge, and that will reverse the level of cyclic AMP or reduce the level of cyclic AMP, and then you will see that the cell can move into metaphase, anaphase and telophase. Now a question comes, how many primary oocyte you have at birth? How many primary oocyte you have at the 5th month of intrauterine life? So questions are asking 5th month of intrauterine life, how many primary oocytes are there? You say maximum. Maximum means it is around 7 million. So there are 7 million primary oocyte in the 5th month of intrauterine life, but they were degenerating. Now if you say they were degenerating, then how many are left at birth? At birth, actually, we are having around 1 to 2 million. So 1 to 2 million only. Yes, and they are still degenerating. If you say they are still degenerating, then how many will undergo ovulation then? So we have maximum 7 million in the 5th month of antenatal life, and they were degenerating, so 1 to 2 million at birth, still degenerating. The total number which undergoes ovulation is only 400 to 500. So only 400 to 500 primary oocytes will be undergoing ovulation. That you have to remember. What do you see in the testis at birth? If we are looking at the testis at birth, then we'll have only the primordial germ cell. See, this is the testis at birth. And in the testis at birth, we'll have only primordial germ cells. Why? Because in the male, primordial germ cells remain dormant till puberty. So spermatogenesis begins after puberty. Now if you mention like that, how much time does it take for the primordial germ cell to become sperm? It is 74 days. It takes 74 days for spermatogenesis to occur.